think I'll start the session today. Just let me know if my screen is visible and whether my voice is clearly audible or not. Can anyone let me know whether everything is working fine?
Yes, sir, it's fine.
Okay, thanks a lot. Yeah, so again, I welcome all of you to the course Bioinformatics Algorithms and Applications. Today we will discuss about the questions and some of the concepts that Professor Pomiha went over in his week 7 lecture. And this is usually not the exact schedule that that we basically follow for this class. So my class was scheduled for Monday, on which I was traveling for some visa-related work, so I had to reschedule it to Wednesday. But anyway, you guys probably have gone over the TA session on the Tuesday's lecture as well. So can anybody remind me what Professor Pomiha went over in his week 7 lectures? In the earlier lectures, we talked about the different aspects of structure visualization, PDB, and concepts and problems related to those, right? So in week 7's content, what did Professor Pomiha primarily focus on?
Sir, actually, I have a doubt from a previous lecture. So before we start on previous lecture, sir, what happened was the question that that was, I guess, for solvation layer, something kind of that. So that question was repeated by Soma sir also after on Monday lecture also and Tuesday lecture also, but the answers of contradict each other. So they were complete opposite. If you will show me the question, I will recognize the question. It was about how the solvent accessibility of the amino acids were according to the distance. That was the question.
So you are talking about a question in the NPTEL platform, is it? Or
no. So your session previous, your previous session. Yeah, this one. Yes.
Yeah.
This exact question was, yeah, I guess.
Yeah.
This one was.
So yeah, so basically, in this particular question, what it's asking is that the solvent accessibility. So solvent accessibility usually means that that particular portion of the protein that has to be exposed to a solvent, right? Just a little bit. So if it is exposed to an external solvation layer or a water kind of environment, so that will mean that these residues are accessible to solvent, right? Now here it is giving values for multiple of these residues. For example, alanine 7, GLU 8, valine 9, sp10, alanine 11, and the values are given in angstrom square. So this is most likely solvent accessible surface area, right? Now, when you talk about solvent accessible surface area, what's happening is that the residues that are located at the interior. So what does interior mean? Interior usually means buried positions, right? So interior will mean that these residues are in a buried position. Okay. So if the residues are buried, so that means that they will have less access to the outer solvated environment, right? So that means for whichever residues the lowest solvent accessible surface area value was observed, so those residues should be present in interior. Okay. So for example, in this case, 0.2 and 1.2, these two values seem to be the lowest among all, right? So 0.2 is corresponding for alanine 7 and then 1.2 is corresponding for valine 9. So residues located at the interior technically should mean that they are in a buried position, and the lower the value of solvent accessible surface area, the then those residues should indicate that they are in a buried position. So that's what the question was exactly asking, kind of, if that makes sense.
Sir, sir, the doubt is that if 52 and 75 are higher, distance, right?
Yeah, yeah.
So the larger the distance, the more the buried amino acid
No, no, no,
is.
no, 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 no. I think you are confusing these particular values with distance. So these are not
Yes.
distances. These are surface areas. If you notice that the unit is in angstrom square, okay. So the angstrom square
Okay.
indicates an area, the area around of the residue, the area of the residue that is exposed to solvent. Okay. Now, if a very little amount of area, or if a very small value of area is exposed to the solvents, then that means that that residue would be present in an interior position. Okay. So this is not
Okay.
distance. This is an area value.
Yes, sir.
Yeah.
So I think so that's from my fault because I didn't remember the unit. So I got
Uh,
confusion Armstrong and Armstrong square.
no, no, no.
So
It's
this
fine. It's
is the
fine.
surface area.
Yeah,
Okay,
yeah, yeah.
sir.
So
Thank you so much for
yeah. So look into
clearing this out.
the parameter called SASA. Okay, SASA is the solvent accessible
Okay.
surface area, and that usually indicates the area of the particular residue, or you can take, for example, a whole residue range that is exposed to the outer solvation layer or solvated environment or not. So if the SASA is of a very high value, then that means that that is exposed to solvent. If SASA is of low values, then that means it is in a buried position. So that's exactly what
Okay, sir.
this question was asking. Okay.
Okay, sir. Thank you so much, sir.
Yeah, it's fine. It's fine. Just camera can block some. Let me just re-enter the meeting. Yeah, sorry about that. I'll just share the screen again. Yeah, so hopefully everything is working again now. Yeah, so in this particular week's lecture, we will go over different concepts that kind of proceed from what we earlier talked about regarding PTB structures and all. So I believe in this week's lecture, Professor Kumiha went over different concepts regarding modeling also, right? Protein modeling, homology modeling, loop modeling, those kinds of concepts. So we'll try to go over those quickly as well. And the few questions that I didn't have the chance to go through in the earlier week's lecture, we also covered that in this particular one, right? So I believe just for a quick recap of previous week's lecture, we talked about the uh, concepts such as information theory in GOR method, where regarding particular type of secondary structures and their prominence for particular type of residues, right? So if you have given questions regarding a particular residue for which a overall information along with a secondary structure information is given, then you can calculate the information content for that particular residue for a certain type of secondary structure using the GOR method. So we kind of went over that using this formula here. And then further, we also talked about the different concepts regarding the protein structures, right? The crystal structures of proteins from the database PDB, right? And we noted that in the PDB files, right? Those are text editable files. If you open them up using a text editor, you will see that there are different columns, right? Different columns that correspond to different values. For example, we noticed that there are the coordinate values, X, Y, Z coordinate values for different atoms. There is an occupancy value that shows alternative conformations for certain residues or certain atoms if that is present. Then we also notice the temperature factor or B factor values, which usually talks about whether the protein is whether that particular atom or residue is mostly rigid or whether that is very flexible, right? So we went over these concepts, and then we also tried to, for example, compare different structures. If you have the same structure, or if you have different conformations that has been crystallized for the same protein, let's say the same residue range, for example, then you can check what the overall difference is between the structures using statistical parameters. One of the most common ones being the root mean square deviation, right? So the root mean square deviation or the RMSD is calculated using the reference sort of atoms, or you can say the reference proteins, atomic coordinates with a particular conformation's atomic coordinates that you are trying to calculate the RMSD difference between, right? So this is basically this is quite handy, I would say, when you try to analyze, for example, different conformations together of you, or you analyze the snapshots generated from something like molecular dynamics. Then you have to see the convergence of the simulation, or if you want to just see the structural differences, so on and so forth, for the entire protein ligand complexes, or let's say just a particular region, right? A small region like loops, or some secondary secondary structures like helix or beta strands also. Then you can do so via uh, comparing the RMSD values with respect to the initial structure. So it is quite handy in a lot of cases, the RMSD values. This is the solvent accessible surface area question that we went over. And then we just went broadly over the PDB database and the PDB files, and you notice that alongside the structures, of course, there are different values that are present. Uh, that kind of tells you about the quality of the structure. For example, we learned about the resolution. The lower the value in the resolution parameter, the better the structure is, right? So, for example, in this case, 1.14 angstrom resolution will be much better than a 3.43 angstrom resolution for certain proteins. So, if you want to plan a computational study using some sort of structures that is deposited in the PDB database, then you should look for if there are any better alternatives for that structure. Let's say for the same protein, if there is uh, a structure with better resolution deposited by a different research group for this particular protein, then you might want to look into that, okay, for your particular study, right? So what resolution usually is? So resolution is basically a metric that uh, talks about how good the structure is, what the quality of the structure is uh, for the protein that is crystallized, right? So here it is, for example, talking about that in a perfectly aligned crystals, where proteins, they will share identical orientations, the high resolution data. So remember, the high resolution data will mean that the value for the resolution will be lower, right? That will capture the details in a very fine manner, right? The diffraction pattern. So that will give you a better quality representation of the structure. It will be able to capture the coordinates much better. Now, when there are variations in the uh, proteins that have been captured by the crystallization process, it could be due to local flexibility or motion, or the diffraction pattern where there is fine conformation lacking. So that will, of course, result in a lower resolution structure. So that means the resolution value will be higher. For example, let's say 3 or 4 angstrom, something like that, right? So it's essentially a metric for the level of detail in the diffraction pattern. Uh, if you have a very high resolution structure, you will see that their crystal, basically the crystallization process, they will have highly ordered arrangements. However, if you have a very low resolution structure, so you will see that uh, they will not be that kind of highly ordered arrangement in the crystallization process. And for those kind of structures particularly, you will need to go for probably for further optimization to be able to use them as starting structures for some sort of study, right? Yeah. So here, high resolution structure basically means that the resolutions with smaller values, right? The smaller resolution values, they have high confidence in their atomic positions, right? So, for example, in this case, you see uh, a 1 angstrom structure. If you compare this with the 3.0 angstrom structures, you will see that the electron cloud, etc., the kind of the topology here that is being shown is very intricate, right? As compared to the 3 angstrom structure, where it's just broadly giving you the shape. However, here much intricate details about the distribution of the cloud is kind of visible, right? So in this case, it is talking about the electron density maps, of course, for the structures with a range of resolutions. Here, for example, all of them are from tyrosine residue, right? So tyrosine 103 from myoglobin. Then you have this residue with 2 angstrom structure. There is 2.7 angstrom. There is 3.0 angstrom as well. And if you compare the resolutions, you will of course see that the lower the value, the better quality the structure is. So that's just a visual representation for it, right? Yeah. So we will just expand on the concepts of PDB just in a bit, right? But alongside this, some other concepts that Professor Gromiha also went over. So he went over something called the transfer free energy, right? Now, what is the transfer free energy? It's uh, let's say um, the energy of transferring an amino acid in this particular context of the question from a position of a buried kind of environment to, let's say, surface kind of environment, right? Now, these kinds of energy calculations, and I have talked about it in some of the other contexts as well. So they are kind of dependent on the population uh, of the different situations. So, for example, here in this particular case, it is talking about valine residue, right? And what the population that I'm talking about here is that here it is mentioned 25 valines a total in this particular protein, and 5 of them are residing on the surface, right? So that means 20 of them are residing in a buried position, right? So that is the population difference between, let's say, the buried valines versus the surface exposed valines, right? Now, here the question is asking you have to calculate the transfer free energy for valine, right? So how can you calculate that? So transfer free energy you can calculate in this particular formula, and any kind of free energy difference calculations that you can do based on some sort of populations, right? So you can use this formula quite handily. So it is minus RT ln f of I. Uh, Now, in this particular case, the f of I that will indicate a fraction, which is how many valines are present in an interior position. So that would be in a buried position. Divide that by how many valines are present in an exterior position. So exterior will mean surface exposed positions, right? So if you plug in the values of minus RT ln f of I would be the fraction, then you should get the transfer free energy value. So can you calculate quickly what this value could be? So we also looked into similar examples of, I believe when I showed you an example of free energy surface, right? When we have to basically calculate a free energy kind of landscape or map of the different conformations arising from MD simulations. We can use the exact same kind of process. Okay, so you can divide the entire data into let's say several beans. Beans are kind of short intervals of the data set. You can imagine of them. You can imagine them as some sort of kind of box, right? And in 100 boxes, let's say your different values or data are plugged in. Now you want to calculate which bean has the most population based on the number of snapshots that are present. That has the corresponding values of the particular reaction coordinate you are using. And then you can compare that uh, particular population of the most populated bean with the other kind of beans which have lower number of population, and you can calculate a relative energy difference using this kind of formula, right? So that will, if you calculate this for the overall kind of MD snapshots, you will get a free energy landscape kind of map. Okay, so that is also quite handy if you want to explore the conformational deviations of proteins in different cases, right? It's quite handy in a lot of other calculations also. So this is the same kind of formula here. I see an answer A in the chat. Okay, so basically what you have to do is that you have to plug in the value minus RT ln of the number of valines in the interior position or buried position divided by the number of valines in the surface exposed position. Now, since there are 25 halines and 5 of them are on the surface, that means that 20 of them are in a buried position. So essentially, it would mean minus RT ln of 4, right? 20 by 5, right? If you plug in the values, so minus RT is of course minus R is R is your 1.987, and that is calorie per mole per Kelvin, the unit. You multiply that by T. T is the absolute temperature, right? 300. The Kelvin gets cancelled out. So your overall unit you are getting in calorie per mole. That will give you a value of, I think these are kind of repeated, but anyway, that will give you a value of minus 8 to 6.37 calorie per mole. Now remember that this particular unit, this is in calorie per mole, whatever you are calculating, right? So for the options here, minus 8.8 to 6.38, option A. So the unit here is different. It's in kcal per mole. Okay. So to convert from calorie per mole to kcal per mole, you will have to divide the value by 1000, of course. So you will get a value of minus 0.826 or around minus 0.83 kcal per mole. So just be careful about the units that are given on the questions, because some values could be different, right? It of course depends on whatever value you are plugging in particularly. So if you used here a different value of R, of course your unit would have been different, right? So that's the gist here. So in this case particularly, the answer would be option B, minus 0.83 kcal per mole, okay, kilocalorie per mole. So you can try to remember this particular formula, and in any type of free energy difference calculations, this will come in very handy. Okay. Yeah, this is the formula that I was talking about earlier. So for a free energy landscape, the formula for kind of calculating the energy for different beams would be minus kBt ln of ni by n max. So n max is your maximum populated beam, and then ni is the population for a particular beam. Uh, and in all of these cases, of course, the Nmax will have the lowest energy, and that will be your minima or the most stable structures. Okay, so this is also quite handy and quite interesting to know. Fine, right? Now, of course, a big part of what Professor Brahma went over in this week, I believe, is homology modeling, right? So in homology modeling, there is a particular term called SCR. What is SCR? What does SCR stand for here? So in this case, the full form of SCR will be the structurally conserved regions. Now, what the note is particularly saying here, of course, it's saying that homologous protein structures, they have greater conservation, right? And the existence of structurally conserved regions or SCRs in protein families with significant sequence divergence, that is what kind of gives the evidence for it. And how can you identify it? You can identify SCRs involving two or more homologous protein structures by comparing them, right? By comparing them together, and you can check the degree of divergence of those kind of structures, and you can kind of deduce structurally equivalent positions among different structures. So structurally equivalent position will mean that a particular subsequence or particular range of structure from, let's say, your entire protein. So that would be quite similar, structurally speaking, and even sequentially speaking, to a certain degree among the different structures that are present, right? So that those will kind of indicate your structurally conserved regions. So that will mean that they could have certain type of functions. That is why nature keeps them quite in a similar way, right? So those are structurally conserved regions and quite helpful in homology modeling. We will just look into the process of homology modeling a bit later, right? And another, so coming to another kind of concept that Professor Kumar mentioned in the earlier class, which I kind of skipped over. So those were the information regarding contacts, right? Residue contacts. Now, what contacts are? Contacts are, of course, uh, in a 3D space, if two residues are very close by, let's say below certain threshold, then you will say that those residues are forming a contact, right? So the contact means they should have some sort of connections. For example, they could have hydrogen bonding between them. There could be a salt bridge interaction between them, hydrophobic interaction, so on and so forth, right? Different types of bondings or non-covalent interactions could be present. But generally, from a distance kind of standpoint, you can say that below a certain threshold, if two residues in a 3D space are present, then they are forming a contact. Now, what is short range and long range contact? So, can anybody explain what does short range contact essentially mean? So, the contacts are already defined by the closeness in 3D space between, let's say, two atoms or two residues, right? So, then why is the range parameter coming in this? Because it's already kind of below a certain threshold, right? So that's because in the contact map, yeah, so few atoms apart in the 2D sequences, yeah. So here, not only in the 3D space, but we are trying to sort of associate them uh, with that presence or their position in the sequence space, sort of, right? So short range contact usually means that the two residues, they are of course forming the contact. So that means in the 3D space they are close by, but they are also very in a very close position in the sequence space also. For example, one residue could be in the 20th position, and the other sequence, or the other residue could be in the 23rd position, right? So they are like three residues apart in the sequence space. So they are in a close position in the sequence space, and if they are forming contact also in the 3D in the 3D structure, so that means that they are forming a short range contact. Conversely, since the folding of protein is kind of complex, right? So it is very possible that the residue that is present in the 20th position in the sequence is also forming a contact with the residue that is present in the 70 position, right? Position 70 in the sequence, right? Because it's kind of folding in a way that these two residues are coming together in the folded structure, right? That means that the residues that are far apart in the sequence, they are also forming contact by being positioned in the 3D environment in a very close in a very close distance, right? So that is the example of a long range contact. So in this particular case, what could the answer be? If they are forming short range contact, so then which residues could be particularly the part of it? So based on what Professor Gomiha kind of mentioned in this particular case, you have to look at the contact map, of course, if the two residues are forming contact at all, and then their positions, right? If they're kind of present in a very close space or not, in the sequence space or not. So just try to see, for example, ILE4 and GLU8, right? So this is ILE4, this is GLU8. So it's talking about this particular dot, right? Then leucine 2 and arginine 6. Leucine 2 and arginine 6 is talking about this particular dot. Then arginine 6 and isoleucine 7. So 6 and 7, of course, this one. And then serine 10 and leucine 2, this particular one, right? Now here, you have to have certain some sort of cutoff to define whether a short range contact is being formed or not. But in this case, kind of if you go by the process of elimination also, the closest contact that is forming between the closest residues are arginine 6 and isoleucine 7, right? Because you can see a presence of dot in this particular place, right? So since they are only one residue apart, in this case, you can confidently say that they are forming a short range contact. Apart from trying to consider any sort of cutoff between the sequence space distance for this particular residue, right? So in this particular case, arginine 6 and isoleucine 7 would be the most appropriate kind of answer, right? So that is the point of contact map. So here, for example, you can take a look at the contact map for, let's say, protein, and you can also try to deduce what secondary structure they are part of. So for example, if I say, if I take, let's say, the residues of alpha helix, right, an alpha helix of a particular protein. So let's say we do a contact map of that, and then we do a contact map of residues that are present, let's say, in a beta sheet, right? So which of them do you think will form long range contact or longer range contacts? Let's say residues of alpha helix and then the residues of a beta sheet. So in the beta sheet, let's say that there are multiple strands are present, right? Just based on their folding, what do you think? So alpha helix kind of will look something like this, right? And then your beta strands will be something like this, right? These will be connected by different loops, of course. Beta sheets will have long range contact. Yes, beta sheets will have long range contact because in this particular case, the beta strands residues, if you look at it, so the particular side chains that will be in the contact forming range, you will notice that they will have a very high difference or very high distance in the sequence, right? For example, in this particular case, if you take a look at the residues that is present here and a residue that is present here, those will form contacts, right? Because they are close by. However, there is a much larger distance between these two residues if you look at the positioning as per the sequence, right? Whether in the alpha, whereas in the alpha CLX you will see The residues that are very closely positioned, of course, it was like I with I plus 4, they are forming hydrogen bonds, right? So they are in contact forming range. So that means that the lower or the shorter range contacts, if you have to distinguish between them, they will kind of form alpha helical structures, right? Whereas the longer range contact kind of arrangement, that will be considered as a beta sheet. That can be considered as a beta sheet structure in this particular case, right? Of course, you have to evaluate that separately. But you can see some sort of association with the secondary structural patterns with the context map here as well, right? So that was one of the concepts. I believe we went over some concepts regarding the percentage of accessibility. So here the question is asking that the accessibility of residue arginine 74 in extended and folded states at 165 and 90 angstrom square. So again, this is talking about the solvent accessible surface area. So what part of the residues are accessible or exposed to the external solvated environment, right? So what is the percentage of accessibility in this case? You just have to kind of find out using this formula. So here it is saying about the accessible surface area in folded state divided by the accessible surface area in an extended state. Okay. So the two values are given here, of course. In extended and folded, it's 165 and 90 angstrom square. So what would be the percentage of accessibility? Just very quickly. So yeah, so the answer would be 54.55%. So particularly you have to divide 90 by 165 and then you have to multiply that by 100, of course. So that should give you an answer around 54.55%. Okay, that's the percentage accessibility. So these sort of different, these are all kind of fraction related values, whether that be the field.
Sir,
Yeah, yeah.
sir, I have a question.
Uh-huh.
In this question, extended is given first and folded state is given after that, right? So
Yeah.
it wouldn't be 165 as extended and folded would be 90. So if I'm not wrong, I am confused because the extended is given before the folded one and the 165 is given before the 91. So for that logic I took.
No. So that,
Equal
yeah. So in this case, that does not matter. If it's talking about the accessibility, uh, then you have to use this formula only. Okay. So the folded state will
No,
go up.
I'm
Yeah, yeah.
not asking about the formula. Formula is, I remember the formula is
Yeah, yeah.
okay. But in the question, as saying, arginine is in the extended state and the folded state are 165 and 90.
Yes.
So extended would be 165 and folded would be 90. I'm confused about that.
Extended would be 165 and folded would be 90.
Yes, because extended is given first and folded is given after.
Yeah.
So 165 is given first and 90 is given after. So wouldn't it be 165 by 90?
No, because you see, it's folded state divided by the extended state, right? So
No, so,
that's the particular
okay,
formula for
okay,
accessibility. Yeah. Same. Yeah.
okay, okay, sir. Okay, sir. Thank you, sir.
Yeah. In this case, it should be folded by extended only. So just remember.
Okay, sir.
Yeah. Just remember this one. Okay. Could you tell me what is the cutoff distance for aromatic sulfur interactions? So in the PDB database for finding interactions, of course, you have to calculate the distances between certain atoms in different residues, right? You will see, for example, if there is a salt bridge formation, two charged residues will be there. And let's say you have to calculate some set atoms distance between these two residues, right? So if the residue interaction or the distance is between below 4 angstrom, let's say they are forming a salt bridge interaction. Similarly, there will be certain cutoff distance for hydrogen bonding interactions as well. And something like aromatic sulfur interactions, they will have also certain cutoff, right? So in this particular case, remember that for aromatic sulfur, or sulfur pi interaction, you will have a cutoff distance of 5.3 angstrom, which is quite kind of large as compared to the hydrogen bonding cutoff distance or the solvent forming distance. But that is the cutoff distance that is generally used in a lot of cases, right? Uh, and alongside that, of course, since we're talking about structures, uh, for classification of proteins based on the secondary structures, Professor Promiha mentioned two particular database. One is SCOP, which is Structural Classification of Protein Database. Another one is CAT, Class Architecture Topology Homology, right? So these divide the different protein structures based on their kind of their secondary structure or the dominant, the secondary structure that are present there, right? So we'll just take a look into the type of classes that have been defined here. So in the previous question, it's particularly asking about the cutoff distance for identifying aromatic sulfur interactions. Okay. So in this case, a particular set of value 5.3 angstrom that is given here. I believe this was the question for one of the previous iteration of the particular course. So that's why I had included here. So here it's talking about the aromatic sulfur or the sulfur pi interactions. Okay, so that's cutoff distance of 5.3. Here you have as per the particular kind of visual here. So you have to calculate a center of mass distance between the aromatic ring, let's say, and the sulfur atom for this particular residue. Okay, so this distance can give you the aromatic sulfur interaction, and based on that, you can find out whether this particular interaction is present or not. Okay. Now, how do SCOPE and CAT databases they uh, divide the structures into different structural classes? So they do so in terms of the dominance of the different kind of secondary structures that are present. Right. For example, in this question, it is talking about certain classes. For example, all alpha, all beta, alpha plus beta, alpha by beta. Right. So all alpha classes, of course, based on the terminology, you can see that these proteins will mostly be dominant in alpha helical secondary structures, right? So you will see alpha helix all around the place in this protein, right? In the case of all beta classes, you will mainly have the beta strands or beta sheets that are present in the structure. Now the confusion sometimes comes between the alpha plus beta and alpha by beta uh, class of secondary structure based classifications, right? So here in this question, it is asking about alternate alpha helices and beta strands which are segregated from each other. Now, what does segregation means? Segregation in this case usually will mean that uh, multiple alpha helices as well as multiple beta strands, they are present consecutively, let's say, uh, as opposed to, let's say, one alpha helix and one beta strand, and then another alpha helix and another beta strand. So in a particular case, for if you see a segregated structure, that means a large number of alpha helix will be present at a location, and then after that you will see, let's say, large number of certain number of beta strands, and then after that you will see certain number of alpha helices, so on and so forth, right? So in this case, if you talk about, of course, the all alpha proteins and all beta proteins, they are mainly dominant in alpha helical and beta strand rich structures, right? But in this case, remember that the segregation or the segregated term was for alpha plus beta class. If you look at the example or kind of the visual of that, you will see that in the case of alpha plus beta proteins, you will see multiple, more than one, multiple beta sheets or multiple alpha helix that are present one after another, right? And then below, before that or after that, you will see again the other sort of secondary structures that is also present one after another in a kind of clumped fashion, right? For example, here in this case, you will see that three beta strands are present, and after that, multiple alpha helices are also present here, right? So these sort of structures will indicate segregations. Segregations means a large number of similar type of structures are clumped together. However, there will be some sort of alternating fashion. For example, one clump will be alternating after another clump, so on and so forth, right? If you compare that with the alpha by beta structure, you will see uh, something pattern like this. For example, one beta strand is here, after that another alpha helix is there, then one beta strand, one alpha helix, one beta strand, so on and so forth. So in this case, there is no clump of beta strands or clump of alpha helices that are present in a particular let's say location or position. So that means that they are not segregated. Okay. So that will constitute of the alpha by beta structure. You can go to this particular link and you can do a bit, you can get a better visualization of these kind of secondary structure classifications also. Okay. Uh, of course, in PDB structures, we talked about the different parameters such as the B factor, coordinates, the positioning, right, so on and so forth. There is also something called occupancy, right? So if you remember one column where in most of the cases the values for certain atoms or particular atoms will be 1, right? So that means that in the terms of occupancy, that particular atom has a very clearly defined coordinate that has been captured. However, it is less than 1. If it is less than 1, for example, 0.95, let's say, so you will usually see that there are, let's say, two values, another value for this particular atom, another coordinate value, for which you will see a certain value of occupancy. If you add these two together, they will give a total value of 1. For example, let's say 0.95 and 0.05 occupancy value for one, let's say, nitrogen atom in a particular amino acid residue, right? So these values where the value is less than 1, this particular in this particular atoms, so that means that they have alternate conformations. Okay, alternate conformations assumed by the residues for this particular atoms. That's why you are getting different values. But if you add the occupancy values of all of those together, you should get a total value of 1, right? So that's the alternate conformations and occupancies. And usually, if you do some sort of studies, you have to process the PDB files first. In that case, generally what the different tools they do is that they remove the alternate conformations where a much lower occupancy value is seen, right? They will mostly keep the, let's say, the large occupancy value, for example, 0.95, if that is present there, right? That was one thing. The concept of entropy we also went over, of course. We know that it's the state of disorderedness, right, or the measure of disorderedness. So, of course, if you compare the different sort of states of matter, solid, liquid, gas, you will see that the solid will have the lowest sort of degrees of freedom, right? So that means that the solid structures will have the minimum entropy. We also went over the concept of entropy regarding protein folding as well, right? So here in this particular case, you will see that the solid, of course, has the lowest degree of freedom as compared to the liquid, and the gaseous states will have the highest degrees of freedom. So that means that the highest entropy will be attributed to the gaseous state, and of course, the change of entropy can be indicated as per from which state to which state the change is kind of being seen, right? So can anybody tell me what is the importance of entropy uh, in a protein complex formation or in a protein ligand complex formation? So does any formula come to mind that can state if entropy is included in the total uh, in the terms of energy, if the entropy is included in the total complexation kind of factor or not process?
Sir, can you repeat the question? My internet connection got lost.
Yeah. So I'm just asking that does any formula come to your mind regarding the inclusion of entropy as a term in a protein ligand complexation, let's say? So in terms of energy calculations, I'm talking about. So for energy calculation or let's say energy of complexation, we usually calculate a Gibbs free energy value, right? So that is indicated by something like del G, right? So is an entropy term included in this particular calculation here also? Yeah, in MMPBS values, for example, as I see in the chat. So for Gibbs free energy of protein ligand complexation, you have a formula something like this. Okay, let's say del G equals to del H minus T delta S. Okay, so what is this delta H value? So this delta G is your Gibbs binding free energy, Gibbs free energy, and then this delta H will give you the value of the enthalpy or the enthalpy change, right? And this particular term minus T delta S, that is your entropic contribution towards the binding. So accounting for all of this, you will get the overall binding free energy value, which you can compare between different systems to get a kind of a better look at which is a better compound or which protein complexes are binding more favorably, so on and so forth. Okay, so there is a huge contribution of entropy uh, in calculation of these types of complexation processes. For example, protein ligand protein complex formation. So as such, it has a huge contributing. It is a huge contributing factor in drug discovery research also. Okay, and usually when we do run simulations and those sort of calculations, right? Somewhat, I think you mentioned MMPBSA in the chat. So the MMPBSA values usually they give the energy value in terms of enthalpy. For additionally, if you want to calculate the entropy values, you have to do some sort of process, for example, like normal mode analysis. In that case, you will find the entropy value or entropy contributing factor for the protein ligand complexation. So accounting for all of that, you will get the binding free energy. Okay. So look at one example later, I believe. But yeah, so uh, that's the energy part. Now coming back to the structure part, let's say. So what does a protein structure kind of tell us about? It has the information of the coordinates for the different atoms, right? And as I just said in the earlier sort of question, uh, to find out the particular interactions that are being formed between different residues, right? You have to calculate distances between different atoms, let's say, of different residues, right? And a lot of in a lot of cases, what you do is that rather than calculating the distances considering a particular atom, you do an overall calculation of a center of mass distance. Let's say you're trying to find out the binding pockets distance to a ligand small molecule, right? So of course, a small molecule is consisting of multiple atoms, right? Let's say 30 atoms, 40 atoms are there. uh, all of the residues that are part of the binding site, they will, of course, each residue will have certain number of atoms. So all of the residues together, they will have a large number of atoms, right, together. So if you want to calculate the distance between the ligand and the binding pocket, uh, then what can you do? You cannot take a single particular atom into consideration, right? Only a particular atom into consideration. You have to take a center of mass distance, a center of mass calculation for the ligand, also for the particular binding sites or the residues constituting of the binding sites for the protein. And then you can calculate, if you get the center of mass positions, you can calculate the distance between the protein and ligand in terms of the center of mass. So that's the concept here, right? So for center of mass, of course, you don't have to usually calculate this by hand. By the way, there are different tools to calculate those. But just for simplicity's sake, here the question is asking for only 4 atoms of a glycine residue. Of course, these are the backbone atoms, right? Um, for the glycine residue. So here you have to calculate the center of mass because glycine is the simplest amino acid and does not have the side chains, right? There is H for side chains, but we are not considering the hydrogen here. We are only considering the backbone atoms here. And using the formula of something like this. So for each of the atoms, you have been given the atomic mass here. So the carbon, nitrogen, and oxygen, they have atomic mass of 12, 14, and 16. Now you can plug in that particular atomic mass here in the MI's case, and then you will have the X, Y, and Z coordinate that have been given here in these 3 different columns, right? So with that, for example, if I give you the X coordinate for the glycine residues solved kind of answer, so what you have to do is that you have to multiply, let's say the first nitrogen, you have to multiply its atomic mass of 14 times its X coordinate, of course, first 6.3. Then you have to plus that with the next atom, right? The next atom is carbon. It's C alpha atom. So that has a atomic mass of 12, of course, right? So it will be 12 times 6.1. So this particular value here. And for the next one, again, it is a carbon. So that is the C here. So it will be 12 times 7.4 because 7.4 is the X coordinate here. And then the final oxygen, you will have 16 times 7.4, right? Divide that by the total atomic mass, which is 14 plus 12 plus 12 plus 16, you will get a value of 6.83. Okay. So now there are two 6.83, it seems here. So the A option and D option also. Could you tell me which option it could be among these? So you have to take into account all of the x coordinates that are given here. For each particular atom, you have to multiply their atomic mass with the x coordinate. You have to sum all of them together, divide that by the total atomic mass of the residues that you are considering, and then you can get the answer. Okay. For the x coordinate, it is 6.83. Right. So for y value also, let's say, I'll just show you the answer. What you have to do is that you have to only focus on the y coordinates, of course. So the first one is the nitrogen. So that means that you do 14 multiplied by 4.5. Okay, I think there was some. Okay, so 14 multiplied by 4.5. Yeah, yeah. 14 multiplied by 4.5, and then next you do for carbon. So it is 12 multiplied by 3.5, of course, right? In this particular case, and then 12 multiplied by 2.8 because another carbon is there, and then 16 multiplied by 1.6. Okay, divide that by, of course, the total atomic mass, you will get 3.04. And then for the z coordinate, again, you can just refer to these particular coordinate values, and you can get the answer in the same way. So just remember the simple formula of summation of mi xi divided by summation of xi. Summation of mi. You can calculate the center of mass. In routine analysis, you don't have to calculate anything by hand. There are different tools assigned for calculating center of mass distances or center of mass positions, so on and so forth. Right. So in this case, the answer would have been answer D. Right. Uh, Now for homology modeling, as Professor Gomeha went over, so if you plot the number of aligned residues, you have to do sequence alignment for homology modeling, right? If you plot the number of aligned residues with the percentage of identical residues, you get a plot like this. Okay, here. Now here the entire kind of set or the entire space is divided into two zones. One is called the twilight zone, another is called the shape homology modeling zone. Okay, so if you compare the aligned residues with the sequence identity, uh, you can get this sort of plot, and whenever your entire kind of range is kind of falling between the shape homology zone area, so for that you can proceed with the homology modeling without any issues, right? So here, for example, it is asking about 100 residues are aligned with 30% sequence identity. So 100, if you compare that with 30, it should be somewhere around this. Okay. Uh, so not quite in the safe zone. It should be somewhere in the twilight zone. That means that you cannot proceed with the homology modeling in this case. Okay. So the safe homology modeling zone, you will be provided this kind of graphs if such questions are coming. But just remember the shape of the curve. Okay. That's one thing. I will just talk about this later. I believe that we should, since we're talking about homology modeling itself, we will just focus on the homology modeling part right now. Okay. So, yeah. Okay. So, homology modeling, what it does, of course, if you have uh, very high sequence similarity between, let's say, different proteins, and one of the protein you have the structure known, right? Some research group has already crystallized the structure and deposited in the PDB database, right? So if you have an unknown, if you do not know the structure of a homologous protein, but you have sequence for that, so what you can assume is that since there is a high degree of sequence similarity between these two, uh, then what you could do, you can get inference regarding the structure from the already known structure of the homologous protein, right? So that is kind of termed as homology modeling here. And of course, for that you have a template and as well as a target. So for the target, you are trying to predict the structure. For the template, you already have a known structure, right? If these two residues are identical in a homology modeling, that's what you can do. Of course, um, in the case of all of this, you always copy the backbone information, a backbone atomic information from the template to the target, right? But here, it's talking about all of the residues are identical. I mean, the template and the target residues are identical. So if the template and the target residues are the same, can you copy the side chain atomic coordinates as well in the homology modeling process? So that's what the question is asking. So just remember that the main chain atoms, of course, those coordinates will be copied, but whether the side chain atoms coordinates can be copied in this case. And the answer should be yes. If you have the same sort of residues that are present in a position, right, in a template and the target, then you can copy the atomic coordinates for all of those. Okay. So you will get to know it better when you talk about the entire process of homology modeling. Now, what homology modeling essentially is, it's also called comparative modeling, right? So comparative modeling means that by comparison, you're trying to build structure. By comparison with what? By comparing with another homologous protein that is already quite known, right? So basically leveraging sequence homology with established structures, and you are trying to predict the structure of some unknown, let's say you are trying to predict from sequence the structure of some protein that is not known, right? And the basically concept is, as I told earlier, if there is significant sequence similarity, then they will likely have similar three-dimensional structures as well. That's the kind of concept here. Similar three-dimensional structures or comparable 3D structures for the homologous proteins. Okay. So if one protein sequence is known, you can confidently transfer the structure to another protein. And basically, the entire step, if you talk about, first comes with target sequence selection, of course. That's quite important because based on so you have a target sequence uh, that you are trying to predict the structure for. And then what you have to do is that you have to carefully select a template for which you will need to build the structure, right? Because you need the homologous sequence. So which could be a correct template that you have to identify using a database search. And then when you have a template in hand, what you can do is you can further refine the sequence alignment, of course, because by aligning only you will get to know that, okay, this particular residue or this particular residue danger structure can be attributed to this particular section of the unknown protein, right? Unknown protein structure. Sir, can there be change in side chain due to rotamers? Yes. So the side chain libraries can be changed. So you were talking about the previous question. So in homology modeling, what they do in general sense, if the residue is same, of course, they just copy the coordinates for the side chain residues also. But for optimizing the structures, as you correctly are saying, because its surrounding environment could be slightly different, right? So that means that there could be some structural changes, different rotamers as well, so on and so forth. For that, you have to further do optimization of the particular structure. Okay. So by optimization, I mean you might have to do energy minimization, to remove any sort of static classes, so on and so forth. So a homology model build structures, you can check it with different tools, for example, ProCheck and other tools, to know if all of the parameters are quite aligning well. And if you do not have a good structure, if there is some sort of confusion or there's some doubt regarding certain parts of the protein that it is not modeled very well, what you can do is you can do certain optimization steps or energy minimization steps. But before that, for building the structure, for homology modeling, it copies the exact same recipe information. Okay. Yeah. So for refinement of the sequence alignment, after that, you will do a model framework building. So this will give you the fundamental idea about what the protein will look like. And then, of course, after that, you will have to add the loops and the side chains. Then this is a good important part, which is the model optimization. And after that, you have to evaluate the model, right? Now, if you go over these points kind of individually, you will see that, of course, template selection, that is the first step in homology modeling, because once you have a template, you will carry on with that for the entire modeling process, right? So it will involve searching for the protein data bank for homologous proteins with known structures. It will have some sort of alignment tools such as BLAST or FASTA. And then there are some sort of guidelines, for example, with at least 30% sequence identity, and then at least 20% of them can be called as safe zone, right? But the main point is that you have to select the template carefully because structurally similar proteins that may be identified, you will find a lot of hits for your template search. Uh, Choose the one with the highest percentage identity while considering for the resolution appropriate cofactors whether those are present or not. Okay, so all of this can has to be included in your template search, right? But after the template, you will have to do something called sequence alignment, right? So sequence alignment means you will align the full length sequence of the template with the target proteins, and then you have to do refinement of the sequence alignment and realign. You have to do realignment of the sequences using some sort of algorithms. Right? Dynamics will help in that optimization. Sure. Dynamics will also help in that optimization. Um, if your kind of dynamic steps are converged, then you can say that the structure is quite stable in that case, right? So usually before any sort of dynamics, you will also do energy minimization steps. So those will take care of the initial sort of static classes that could be possible in the structures. And then after that, in your dynamics data, you will see if your system is converged, then the structure is likely a stable one, right? So that will also help, certainly. Project can give different results after solvation and before. Project can give different because I believe that due to solvation, you will see certain sort of environment change in the proteins, right? So it's better that if you take the project structure uh, for further kind of optimization, if you run dynamics with a steady kind of solvation state, for example, in your target sort of environment, let's say you are trying to go for a drug discovery, right? Uh, and in this case, you could take like 0.15 molar salt concentration in a solvated state. So that will kind of take care of the external environment, and in the dynamics result, you will see what sort of structures are kind of arising, right? So that's kind of the best approach here. Of course, for a lot of cases, for small scale studies, you really where you really cannot do dynamics, then you have to go for some other options also because it's computationally expensive, so you cannot always go for it. Um, yeah, but that's okay. Usually, a lot of tools that you use for stuff like docking also they will have also their protein preparation steps. So that also take basically care of certain parts regarding the uh, structural model of the protein. Okay, so that will also help for the initial refinement of the structures in those cases, right? Yeah. But yeah, so you have to do the sequence alignment. You have to refine the sequence alignment, uh, and then after that, what you have to do is you have to do backbone modeling, right? Now, when you have a alignment for your template as well as your target, then you know which sort of residues corresponding to the template is related or can be associated with which particular residue of the target, right? Now, what you can do is you can copy the coordinate of the corresponding residues from the template onto the target protein, and that will give you the core of the protein in the terms of their backbone, right? Now, if the aligned residues are identical, just what I talked about earlier, then what the process does is that the main chain and side chains atoms coordinates, all of them are copied. Okay. But if they differ, of course, only the backbone atoms are copied for the backbone model building step, right? Then after that, you have to do the side chain atom building. Uh, Usually, for simplicity's sake, or a lot of times, single template structure is used. And then multiple template structures are also viable, but you have to also look at if the computational cost is increasing or not. But you have to kind of carefully take into account all of the choices here. Okay, now if the residues are different, then what you do is that we'll talk about it. You will use the different rotamer libraries for building the sidechain atoms, right? Now, in a lot of cases, you will see that of course you are building the backbone, right? You are trying to build the backbone of the protein, but let's say in certain parts the backbone structural information was not present in the template. So that can happen also, because for the residues that are highly flexible, what can happen is that the exact coordinates for them could not be captured, let's say, in the extracrystallographic process, right? So there is gap or there is hole in the model in that case, in that sense. Uh, and these particular regions, most prominently seen in the case of loops, because loops are unstructured regions, and a lot of times they do not have defined structures for certain residues in the loops, in the PDB structure, right? So that will necessitate a loop modeling step, which is a challenging aspect because you have to sample a lot of there's a phi angle orientation, so on and so forth, different parameters for the residues corresponding to the loop region which are missing in the structure. Okay. Now there are two methods for loop modeling here. It is talking about database searching method as well as the ab initio method. Now what the database searching method is, it will find in the PTP database, let's say, suitable segments from other known proteins that you can fit into uh, the region of the loop which needs to be modeled. Okay, so that's simply the database searching and kind of plugging in the values for the particular residues in the loop that are missing from other known proteins in the database for the PDB database, right? Uh, Another method which is the ab initio. So ab initio means using the first principles, right? So that is a more computationally expensive process where you might have to take into account, you might have to sample basically a lot of structures uh, for the different parameters, let's say phi torsion angles. Then you might have to do energy minimization process using force fields, so on and so forth, right? So that is usually from the first principles, from the physics-based force fields, to try to minimize different static classes, so on and so forth. But if you do not have the computational resource for that, it could be very hard. Okay. But once you uh, sort of find the particular orientations of the loop, then you can plug in into the missing region and you will get the particular backbone of your loop from that. And after that, yeah, basically what it is saying, the same thing. Uh, After that, you have to go for the sidechain refinement, right? So what the sidechain modeling looks like, that will be done using different rotamer libraries, right? So the rotamer libraries, what they are particularly preferred sidechain torsion angle profiles or libraries that are also derived from the known protein structures, protein crystal structures. So based on the different libraries that can be the different torsion angles or the rotamers that can be present for certain residues, you can sample all of that. You can plug in, and then you can see whether certain type of profiles, torsion profiles are giving rise to steric hindrance or not. And based on these different types of samplings, you will find the optimum, uh, let's say, the rotamers for the side chains of all of the residues in your protein. And that will take care of your side chains, basically, essentially, overall, right? But here it is saying that further optimization can be necessary to minimize steric overlaps with the rest of the model structures, which is quite important, right? Uh, for example, as you told earlier, that there could be uh, optimization via dynamics, there could be optimization via energy minimization, so on and so forth. Okay. Yeah, we have a multiple sequence joined by linkers, and all of them are known, but the linkers can cause disorder. Will this be carried into the will that be carried out ab initio or template will be used? So you have a design sequence. So that means that this is a novel sequence that is for a novel protein that is not known. And okay, and you have joined those sequences by linkers, and all of them are known, but the linkers cause disorder. So the linkers cause so is this some sort of like vaccine development or something like that? Just asking for context. Okay, yeah. Okay. Mm, Yeah, so you have basically taken, for example, different epitope kind of structures and then, yeah, antigen development. Okay. Yeah. And then you have joined them by linkers, and the linkers are causing disorders. So disorders in the sense that in your particular model that you are building, KK linkers. Okay. So you are for modeling what you are using, AlphaFold, or you are using homology modeling or anything like that. Rootnix, AlphaFold. Okay. But you are not getting the expected result because your linkers are causing all of the regions that you are trying to join together. It is causing disorder. Homology giving very low scores. What does alpha? What is AlphaFold? Okay, as well as all of as well as AF3. AlphaFold 3 is also giving low score. I guess in that particular case, what you can do is you can start with the AlphaFold structure, and then you can do a dynamics of that to see if the structure is stabilizing at certain time range for simulation. But which like how many total number of residues if you have? If it is a lot of residues, then it could be 150. So 150, I think you can do dynamics. So I would say start with something like the AlphaFold structure, and then you can go for a dynamic simulation up to let's say 500 nanoseconds, something like that. Okay. Then if you see the structure is becoming quite stable, I believe that that will work for your further process. 9 epitopes joined by 8 linkers. Okay, okay, I see. Also, another thing you can try. Also, you can try the Rosetta Fold suit also from Baker's lab, David Baker's lab. Uh, that can be also an alternative for AlphaFold. Bolts 2 I haven't used actually, so I cannot tell exactly how good it is. There is another tool. I'm just not remembering it. So does Pipfold work for this type of design, for structure design? I have to check what is the residue length limitation for that as well. Is mentioning linkers coordinates as modifications useful in this case? The linkers coordinates as modifications, for example, in the AlphaFold servers or in the AF3 software, or no idea, haven't used Pipfold. Okay, okay, I see. Yeah, maybe you can try that as well. But I would say the best is that if you are not getting the particular result that you are looking for from the AlphaFold structures as well, so just take the AlphaFold structure initially, do some energy minimization and run dynamic simulations. And then after that, you will see that if the structure is kind of converging, that then you can proceed forward with that as well. Yeah. So yeah, after that, you have to do some sort of energy refinement using different functions or algorithms, right? So energy minimization will usually mean that the atomic positions will be recalculated, and then based on whatever, whichever case you get the lowest steric hindrance or the lowest energy potential. So that will mean that that is kind of an overall better structure, right? Without altering, here it is talking about without significantly altering the overall structures. So the aim is to relieve steric collisions and strains, right? So that is the idea behind using the energy functions for minimization process. Okay. Yeah. So and then after that you do the model evaluation. For example, you check the phi psi angles, the bond lengths, the close contacts, so on and so forth. One software, as mentioned earlier, also is Project. You can check your model structure here, and if necessary, you can do further optimization of the structures as well. Now, what I wanted to quickly give a demonstration for, I'll just stop presenting maybe from here. But what I wanted to quickly show you, not the entire homology modeling part, but one associated part of the homology modeling, which is the loop modeling, right? So if you remember, in our earlier classes, we used one particular tool called UCSF Chimera, okay, right? So UCSF Chimera is a visualization tool for the proteins or for ligands or any sort of biomacromolecules, right? And then in the toolbar of UCSF Chimera, there, if you remember, there were a lot of options for different sort of calculations, right? Apart from visualization. Now here in this particular case, we will take one particular structure from the protein data bank, which is the 2IQA structure. So it's an influenza A virus nucleoprotein with 3.2 angstrom resolution. And if you look at the biological assembly data that is given here, it has three different chains, right? Let's say ABC chain. And if you look even more carefully, you will see that there seems to be some sort of dotted representation at certain parts in the structure. Now, what these dotted sections or dotted segments are saying, they are basically saying missing regions. So if I open up this chimera, open up using chimera only the C chain of this particular protein, which I have extracted earlier, you will see in this particular case for the structure, these regions, uh, there is a dotted kind of representation that is present. It's not showing the exact orientation for the backbone and the side chains. If you visualize all of the atoms here, you will not find that information in this particular crystal structure, right? If you go to the sequence for the sequence viewer, you will see that these regions are highlighted with a red kind of rectangle, right? If you just highlight this particular portion, you will see that this is highlighting exactly one of the portions uh, of the protein where this kind of dotted representation is present, right? So that means that uh, these sort of segments, they do not have the coordinate information for the residues that are present, that are supposed to be present here, right? So in this case, what you will need to do, you will need to do loop modeling. Okay. Now, for loop modeling, we can use one particular tool, search it out. It is called Modeler. Okay, so Modeler is a tool or a software package from the group of Andre Sally, and it is available freely. So you have to just register. You have to give, I believe, information regarding your institute, something like that, and then they will send you in your email a license key for the tool, right? Now, the good part of Modeler is that you can use it integrated with UCSF Chimera as well. So if you go to the action section here, or not action, if you go to the tools section here, you will see that there is an option called structure editing, right? Under the structure editing in the UCSF Chimera, you will see model slash define loops. And if you hover over it in your cursor, you will see that it says use modeler to model structure. So if you go for this option here, it will open up a variety of options, right, that you can select here. For example, you can model the active region, you can model a default chimera selection, you can model a non-terminal missing structure portion or all missing structure. So in our case, let's say we are only focused on the internal part of the protein. So these terminal missing portions, they are not needed. We will go for the non-terminal missing residues modeling, and then it will ask you different options to select, right? It will ask you how many adjacent residues uh, that are just surrounding, that just flanking the particular missing portions do you want to keep flexible. Let's say we have the default option of 1 here, right? That we will select that on. Then how many number of models you want to generate. So let's say we will generate 5 models as per the default. Then it will ask you about the location of the executable. Now in our Linux system that I'm working on, uh, if you go to this particular location, you will see a lot of different executables are present here, right? If you go for the so m value here, you will see in somewhere here you will get the modular executable mod 10.7, right? So for my case, the location was slash usr slash bin, which is what exactly I have given the path for this mod 10.7 executable. Now, if you just run this script or run this particular tool, it will again start processing the structure, and it will start building the particular loop regions using sort of an ab initio kind of method. Okay, so it has uh, the the structural information for the already available residues apart from the missing region, which will provide for the spatial restraint sort of, right? And then for the particular missing gaps or missing residues, it can sample a lot of different torsion profiles, different coordinate parameters that is possible, and then it will build the model structure from there. Okay. Would it be possible for you to share? Yeah, yeah, definitely. We will do that. I'll share it with the NPTEL team, and then you can take a look at it in the YouTube also. Right. Yeah. So it's currently, let's say, running with 14% completion. It will take some time. A lot of cases when you run modular, you can use the type of Python scripts as well. For example, in one of my earlier calculations, the executable was not operating directly from Chimera, but it was generating the .py script for running, right? And I just had to do some sort of modification. For example, adding in this residue range just colon a, so the chain specifier. So that sort of small glitches can happen, but you have to just take care of it using the code manually. Yeah, so these kind of codes will be generated, and then in the output file you will also see the different models. Along with that, the scores for the models that the modeler kind of specifies, those will be generated. Okay, so if I for example take a look at what the possible scoring could look like. So in this particular case, let's say different models are generated, you have something called the Z-dope score. Okay, now what the Z-dope score is, if you go for the documentation, you will find about this code somewhere. So it is talking about, uh, let's say in simple terms, how good the model is. Okay, and if your Z-dope score was below -1, so -1.2, -1.7, -1.8, -2, something like that. So that means that it is a very good quality model in general sense. Here in the particular calculation that I did earlier, I could not achieve that kind of score, so it was only -0.71920. But it's a decent enough score that I can do further optimization, maybe run some energy minimization calculations or run some molecular dynamics to get a better structure. Basically, that's the sort of point here. Okay. Now the calculation that I'm currently running, it is at 42% completion. We'll just see the results in a bit. Okay. Uh, just a minute. Yeah. So I'll stop presenting from here. We'll look at the results later, and then we will come back to some of our other questions. So yeah. So these kind of questions again, as per the transfer free energy of folding and unfolding, just remember this particular value. So between folded state and the unfolded state, usually the free energy change lies between 5 to 25 kcal per mole. Okay. So that's one value that you could remember. And then another one is that in the unfolded state of the protein, which of the following force could be dominant? Of course, we know uh, the entropy, right? Entropy means that the state of disorderedness or the measure of disorderedness. So disorderedness. So in an unfolded state, since there is a higher level of degrees of freedom, you will see that entropy will be very high in this particular case, right? Now let's say I just keep what I talked about earlier in the case of protein ligand complexation. We talked about the Gibbs free energy, right? And I believe that as you all mentioned that Gibbs free energy for protein ligand complexation that can be calculated using something called MMPPSA from a molecular dynamics dataset, right? And what MMPPSA does is that it gives you the different energy values for the protein ligand complex. So it's the complex minus receptor minus ligand value, which will give you the delta, right? The delta meaning the difference. And it is composed of different terms. For example, your van der Waals interactions, your electrostatic interactions. One term is called the polar solvation energy, another is called the non-polar solvation energy. And this is kind of the overall delta, which is your binding enthalpy, right? So binding enthalpy is minus 31.4008 in this particular example, let's say, right? Now, alongside the enthalpy, if you remember, we also had the inclusion of entropy in the overall binding free energy calculation terms, right? And in this particular case, the entropy part, so the uh, enthalpy part was calculated using an approach called MMPPSA, which is called the molecular mechanics Poisson-Boltzmann surface area approach. And for the entropy calculation, again, the same protein ligand complex, same trajectory, normal mode analysis. This particular calculation was performed, right? Now remember, we know that del G equals del H minus T del H, right? So that is the Gibbs free energy. Okay, what we are asking for here. And you have been given this kind of chart for the overall energy decomposition. So could you let me know what could be the Gibbs free energy of complexation from this data? So you have to take into account the entropy as well as the enthalpy part for the energetics, right? So the overall enthalpy is this one. This is minus 31.4008. And the entropy contribution is this one, minus 70.8647. So you don't have to do much. You just have to use this formula, and you will see very quickly. Uh, if you just plug in the values, you will get the answer. Okay. You just plug in the value, you'll get the answer. And the answer in this case will be minus 13.53 kcal per mole, which is option A, right? Uh, so now, let's say this is for a certain protein ligand complex that you are doing the calculation for. Let's say this is compound 1, right? You are targeting some binding pocket of a protein which is relevant to different disease progression. So you are trying to block this particular pocket of the protein, and you have developed or designed this particular molecule or for this particular therapeutic targeting, right? Now you had already a previously known inhibitor molecule, let's say that has been approved by FDA, which is your control, and for that your delta G value was minus let's say 9.37, right, kcal per mole. Okay, and then you have another compound which is compound 2, for which you have the delta G value of let's say minus 8.2, okay, kcal per mole. So which one amongst compound 1 and compound 2 will you propose as a better inhibitor? Yeah, minus 31. Minus 31 because it's the more negative value, right? So the lower the value in this case, I mean the more negative the value in this case, the better is the complexation, or the better is the binding free energy, because it's the energy value of the end product, which is the complex, minus the energy value of the initial product, which is the receptor and ligand separately, right? So if it is having a higher negative value, that means that more amount of energy is being liberated upon complexation. So that means that this complex will become energetically minimized more, right? Energetically more stable in a sense, right? So the more negative, the better in this particular case. Yes, the lowest value. So just one moment, because I think the loop modeling has finished. I'll again try to share from the other screen. But yeah, so hopefully you can see it now. But this is the result for the USSF Chimera integrated modelers loop modeling. Okay, and after the calculation is finished, you will still see. So you will firstly see a lot of different structures, right? The initial one in this kind of grayish color, you will see that the dotted structure is still present, but overlapping this particular initial structures, you will see other structures are also present, for which loops have been built here, right? So these are the different orientations for the loops that have been calculated using the modeler software tool. Now, if you just look at the individual scoring here, so for the individual score here, again, you have generated 5 models, for all of which you have this Z-dope score. And the best Z-dope score seems to be the hashtag 1.5 or the minus 0.75 score, right? So if you just click this one particularly, it will only select this particular model. So as you can see, the different after sampling the different positioning for the backbone, it has selected this one for the best dope score here of minus 0.74, right? So this is the orientation in this particular missing region for earlier, and this is the third one, okay? So the different orientations are selected here. If you go over all of the different calculated structures, you will see that those are different, right? Now, of course, uh, a z-dot score of minus 0.74, it is quite not up to the mark of the threshold that minus 1 usually which is usually used or cited in a lot of cases. But even if your z-dot score is quite good, for example, minus 1.75, let's say, right? In that cases also, you have to go for further sort of simulation steps to be sure that the loop structure that is being generated is not structurally flawed in any way, right? So in this case, you are not getting a very good dope score because, of course, the number of residues that are being modeled or the number of gap residues, missing residues, that was very high. So it still has a lot of flexibility, and exactly what the model or what the actual structure or the native structure could be, that is quite hard uh, to kind of understand or get a hold of, right? So that is why you do not have that high of a Z-dope score. But even in the case of you where you get a good dope score, for example, let's say couple of residues, 3 to 5 residues, in that case also, it's better that you do some sort of optimization and minimization process. Where to learn the details of the software, such as the settings of modeler, other than the official papers, any YouTube channel. YouTube channel, I do not know if there is explanation of such, but what I could say that you can sample some research papers. Okay, so apart from the original research paper, you try to see what the recent research papers that are using this sort of tools, what sort of settings they are using. Uh, I believe in some cases for the research papers, in supplementary information, they will mention a lot of details regarding these sort of parameters. Okay, so from there you can get a good idea about what is the standard. You can also try using the default, and you can try to do different settings. For example, let's say the option for the flexibility, right? So if you earlier tried with keeping multiple flanking residues flexible, for example, 2 or 3, and you didn't get a good Z-top score, then what you could do is that, okay, you keep the number of residues that has to be flexible on the side of the loop, you keep that to a minimum, and that could change the Z-top score. So basically, try to play around with the different parameters and see what sort of results you are getting. And apart from that, what the recent research papers are referring to. So what sort of settings they are using. So I believe that would be a better uh, way to kind of uh, try to go for an ideal setting. So what the recent research papers that are using the software, what they are using basically. Okay, so I believe that will be the best source in this case, and in all cases, essential, because in YouTube videos and such, you will get more like generalized information, not the exact research based kind of information. Okay, but usually the default options that the softwares keep, they are quite good in a general sense. Yeah, so that was about briefly about modeler, and uh, the section for homology modeling is kind of also briefly covered here. Okay, so that's fine. And then apart from that, one another concept that Professor Gomiha, I believe, went over, that was regarding some urea denaturation experiments, right? Now, what these UDA denaturation experiments they do, uh, they will, let's say, measure the fluorescence intensity for the folded state, for the non-folded state, and for certain state in between. And in this particular question also, you have been given the intensity values for those different cases, right? Uh, Now it is asking that again, what is the free energy change of a particular intermediate state from the initial folded state. Now, how can you calculate this sort of free energy change? You have to calculate the fraction of unfolded state as well as the fraction of folded state. And as very similar to the earlier formulas that we have been using, you can calculate the free energy change using minus RT ln K. In this case, the K would be a fraction, right? So how can you calculate the fraction of the unfolded state here? We have to do using this formula. So in this particular case, the Y particularly that is being mentioned, that is the value for the current state. Okay, so the current intermediate state that you have been trying to calculate the value for, and then YF will be the fluorescence intensity for the whole folded state, and then YU will be for the unfolded state. So here in this case, for the fraction of unfolded protein, you can calculate it using uh, YF minus Y divided by YF minus YU. So just plug in the formulas. So for the folded state was 366. If you minus that for if you subtract 256, which is the current state's fluorescence intensity, and you divide it by 366 minus 51. So in this case, 51 is the completely unfolded state's fluorescence intensity. You will get a fraction value of 0.349, right? And for the folded kind of population, you will get the fraction. If you subtract 0.349 from 1, you will get the folded states kind of population, let's say fraction value. And for the case of K, for K, you have to take fraction unfolded divided by fraction folded. Okay. So again, if you plug in the values as earlier, you will get an overall calculation of 371.631 calorie per mole. Okay. You can convert this to kcal per mole using by dividing this value with 1000, right? That is that can be done. So just remember this sort of free energy calculation formulas because we saw the usage of similar formulas in this particular fluorescence intensity, and then in earlier cases free energy surface or in transfer free energy in all of those cases. Okay, so that can be used in different scenarios. So yeah, so that was one numeric question. Uh, but since we are trying to talk about, we were just trying to talk about um, the structures of the proteins, right? And we also talked about the homology modeling. And all of that kind of overall relates to uh, the question of how you can try to build or model an unknown protein sequence, let's say, using different algorithms or tools and so on and so forth, right? And this has been a long-standing question for a lot of time. Uh, Protein folding problem, as what it is being referred to as, right? Now, very related to this particular protein folding problem is one term called CASP, CASP. Do you know what CASP or CASP means? It is something related to solving the protein folding problem. Basically, trying to get the structure of a protein from its sequence only. In that sense, yes, they hold competitions for tools. Yes. So CASP, the full form is Critical Assessment of Protein Structure Prediction, right? What it is basically a bi-yearly competition where different research groups, they will go there with their developed algorithms, and they will try to see if their algorithm can predict accurately the structure of a protein from a new sequence for which there is no experimental structure data available, right? Now, the competition was first pioneered by Professor John Molt and his colleagues from University of Maryland, and you will see that CASP1 actually occurred in 1994, so a long time ago, right? And it's a bi-yearly process. So in CASP2, you will see it's in 1996, then in 1998, so on and so forth, right? Uh, for the longest standing time for this competition, so the organizers, what they do is that, so what they did is that they set a threshold score of 90. That if your algorithm is passing this score, then that means that your algorithm is uh, capable of accurately predicting the structure. And what structure they are predicting? So what they do is that a very recently solved crystal structure, which is not disclosed, not published anywhere, so that is known to the organizers only, and they will only provide you the sequence. And they will tell you, okay, use your algorithm, build the 3D structure of this protein. And then when you build it, they will compare with the actual resolved structure that only they know the information of. And they will see which model is doing in what capacity, right? Which model is better, and comparing of the different models and so on, right? Now, for the longest standing terms, for the longest time, basically, no algorithm or software could reach this particular threshold score of 90. In CASP, I believe 12. So CASP 12 occurred in 2016, right? Okay, so CASP 13 was occurred in 2018. So in CASP 13, I believe. So in CASP 13, what happened is that one particular group, they came with they developed software, and even though they did not have uh, the capacity to reach the threshold score of 90 at that particular time, but their software was very well above the rest of the competitors. Okay, so their software got very close. It could not do the threshold score of 90. It was still quite far from that. But as compared to the other competitors of the same nature, so it performed very, very well, right? And what the developers of this tool basically did is that after CASP 13, they again modified their particular algorithm. They did different tweaks, and they came back in the next CASP, which is CASP 14, and then what they did is that they were able to achieve the threshold score of 90. And at that time, people were talking about that the protein folding problem is solved because one algorithm has emerged that is capably predicting the structure of an unknown sequence, for a sequence that is structure not disclosed, right? So do you know what is the model called? What this particular, yeah, this competition is still being held, I believe. Although in recent news, I believe like a year ago, I saw that the funding for the CASP competition was kind of terminated, or that is in a very kind of awkward position after the different geopolitical kind of things were going on.
Sir, a few years back when I was doing
Mhm.
that, I guess I heard that in this competition, the students who didn't know bioinformatics but
Okay.
was from the
Mhm.
hacking background and computer
Mhm.
science background, they
Okay.
took part
Mhm.
in this competition and they scored better than the people who have actually done the bioinformatics course. It
Oh,
was
okay.
a news at that time. I was
Uh,
reading about that.
so at which particular year it is?
It was, I guess, around the 2020s. I was
Yeah,
doing 12,
yeah, could be.
2020.
So yeah. Because in CAS 14 only in 2020, as you see, if you read the text here, the model that I was talking about, which is AlphaFold 2 at that time, right? So they, yeah, AlphaFold. So they basically could reach the threshold score of 90. So at that time, right? And AlphaFold, if you look at their history, so it is of course developed by DeepMind. DeepMind is a part of Google now, right? And what they did is that they have trained different types of models in the past. If you look at their history, they have also experimented with the different board games also. So, for example, you might know chess, right? You might know Go. So they first, I guess, tried with something called AlphaGo. So AlphaGo is a basically machine learning model or a deep learning model that plays Go at a very high level. And at that time, what people are talking about, so Go is basically a very highly intuitive game kind of. It has a very large board if you compare it with other board games like chess. So Go has a very large board, and there is a lot of permutations and combinations that can happen in the different moves of Go. So that's why brute force calculation is quite not possible for the computer to do in this particular game. And what the AlphaGo project it was able to achieve is that it was able to uh, learn the game by playing with itself different times, uh, of course thousands and lakhs of times, and then after that it could play with the best of the world, right? At the time, the legendary co-champion Lee Sedol, I believe, it is also documented in a quite good documentary in YouTube, AlphaGo the movie. You can take a look at it. But at that time, what they talked about is that the deep learning process or the advancement in deep learning process after the development of AlphaGo, that has been advanced uh, like quite what people could not expect, basically. So the advancement, this kind of advancement was attributed to like a 10-year leap in the deep learning protocol advancement. And then after AlphaGo, they also looked into Alpha Zero. Alpha Zero is a popular chess uh, software or chess playing kind of deep learning model. You might have heard about it also. And then from that, experimenting and doing research on the different board games, they have transitioned to AlphaGo also. To AlphaFold, which is the protein folding problem solver. Um, I believe they have reverse engineered AlphaFold with an open source thing called Protonix. Okay, it has more capability. So yeah, it could be, because I think Alpha Fold, they have made their code open source quite a while ago, right? So different people are doing experimentations on their code as well as the other codes that are available. A lot of very interesting research in the protein folding domain is kind of unfolding now. That's very true. And related to that, of course, you will know that the Chemistry Nobel Prize for 2024 that had been jointly awarded to Professor David Baker. Alongside them, of course, the half of the Nobel Prize that was awarded to Demis Hassabis and John Jumper, both from the AlphaFold project, right? So Demis Hassabis is the CEO and the leader of the projects in DeepMind, and then John Jumper is the first author for the paper for AlphaGo. If you look at it, so he was kind of the lead scientist that was working on the project here, right? For protein structure prediction, they have been awarded the Nobel Prize for chemistry in 2024. The other half went to Professor David Baker. So his work is in computational protein design. So basically, can you design for a certain function a protein that is not existing in nature, right? It can have implications in a lot of cases. For example, in environmental sustainability, drug discovery, medicine, so on and so forth. A huge broad area of application is there in this particular case or the scope of this particular project. As Professor David Baker also says in a lot of his speeches, is that for a lot of problems that we face, if you let evolution take course of that, right, it will eventually probably take care of it, right? But evolution is a very slow process, right? It will take billions of years for that. We cannot wait for that long time. So what we can do is that what evolutionarily still does not exist in nature, we can design those sort of proteins, and then we can implement them in different issues that we are facing. Let's say some enzymes that could degrade plastics, right? These sort of different interesting examples that he goes over, and a lot of very high impact work is being done in his lab. So you can take a look into their different speeches, their Nobel Prize reception speeches, and then their different lab websites and papers as well, to get a better understanding of that. Quite interesting. Okay. So I believe today I will just end on this particular note. Um, I will maybe try to include some of the papers from the AlphaFold group at Professor David Baker's lab also in the shared Google Drive folder. So maybe you can take a look at it, how they do it. Okay. Yeah. So today I will wrap up the class now, and then we will meet in the next week's classes. So the next Monday. And as this class also has been rescheduled from an earlier class, so I think I will have to reschedule the next week's classes, not the next week, the next to next week's classes to some other days. Maybe we prepone it or postpone it. I'll have to ask the NPTEL platform for that, because I'm moving to Japan. I'm just going to Japan for a conference for this particular week, so I will not be available at that time. So we'll just request to the NPTEL platform and see what they suggest. If you can click on or postpone those. Okay. So apart from that, thank you all for joining, and then we'll look into the contents of the other weeks in the next classes. So it will be possible for you to share the video recording. Yeah, definitely. So we will I'll upload the video recording of the session on YouTube, and we'll share with the NPTEL team, of course. Particularly we'll update it on the shared Excel sheet that you guys also should have access to from the NPTEL platform. I believe you have, right? So we'll just update the YouTube link there, and you can take a look at the recorded lectures. Okay. Do you run these tools on dedicated GPU of your own? So for modeling purposes, for example, Chimera using modular simple loop modeling and etc. We just run it on our CPU using simple computers, right? But for running multi-dynamic simulations, we do have dedicated GPUs and high performing clusters and so on in our lab particularly. So for running MD simulations, we use GPUs. If I give you example, for example, NVIDIA 28 Ti or NVIDIA A4000 data generation, those kind of GPUs we have access to for a particular lab work. So for any extensive calculations that you need to do, we need to do. So we run those calculations on the dedicated servers. Dedicated GPUs, we have access to that in our lab. But for running simple loop modeling, etc., you can do it in your own computer quite well. Okay. Okay. So yeah, thank you all for joining. Then I will just wrap up the class today.
Thank you, sir.
Yeah.
Thank you, sir.
Yeah. Thank you. Thank you so much. The course related books suggest. So the course related books, I will say that if you want to take a look into the fundamental concepts, so go over some of the books that I have included in the Google Drive. For example, David Mount's Bioinformatics, or you can perhaps, if you can get a hold of Professor Blomiha's book on protein modeling, you can take a look of that also. But best I would say is that take certain sections and then read related research or review papers on that. Okay, so that will help you to keep up with the recent advancements and so on. I believe that will help the most. Okay. For broad understandings, you can take a look at the old books, for example, the bioinformatics book by David Mount or Professor Pomiha's books, these kind of books. Okay. But to keep up with the recent research, you should look into the research or review papers that are coming out. Yeah. Okay. Thank you, everyone. Then take a look at the Google Drive folder, shared Drive folder. I have included a lot of resources there also. And then we'll meet in the next class. Thank you.